All right. Hi, everyone. I'm happy to introduce today's speaker, Dr. Todd White, to the GT Neuro Seminar series. Dr. White received his bachelor's training in biomedical engineering at Harvard University and his doctoral training in neuroscience at the University of Florida. He completed postdoctoral training at the University of Florida and the Morehouse School of Medicine. Currently, he's a research biologist at the Atlanta VA healthcare system, as well as an assistant professor at the Morehouse School of Medicine. He's a career development awardee from the Department of Veterans Affairs Research R&D. Currently, his research is focused on improving diagnoses for treatment of neurotrauma. He uses preclinical models of traumatic brain injury to better understand molecular and behavioral consequences of injury, with the goal of using this information to help develop protocols for halting the progression of injury and improving behavioral status. Ultimately, he hopes this work will translate from preclinical models to help improve outcomes for people suffering from traumatic brain injury. So he has a wonderful talk prepared for us today, and I'm excited to welcome Dr. White.
Thank you for that introduction. It's a pleasure to be here. So, in my lab, we are basically trying to develop at this time a model of chronic, repetitive, mild traumatic brain injury. And what I wanted to share with you today is basically an update of where we are in this process. And so we have progressed through a couple of different models of this, and hopefully what I'll be able to gain by sharing this information with you is a little bit feedback from you, and also help me move this research forward. So what is the issue? So in addition to TBI being an issue for the civilian population, from the VA's perspective, it's a larger issue in terms of the veteran population. As many as 30% of veterans have suffered traumatic brain injury, and even with mild TBI, the effects of the injury can be detrimental to veterans' quality of life. And so with persistent symptomology, which can include psychological, motor, and visual deficits, excuse me, and this can be especially true with repetitive mild TBI. And so we wanted to find a way to address a situation where a veteran may walk into a clinic and have some symptoms that might be related to TBI, but doesn't really know the origin of those symptoms. And so how do you correctly, diagnose and then treat that patient? How do you determine causality? Dr. Meller, Robert Meller at Morehouse School of Medicine, has been using RNA sequencing to look at stroke and TBI patients, and he's been able to find a blood molecular signature using RNA sequencing, and find a pattern of genes or a panel of genes that could distinguish between stroke and non-stroke, and can also distinguish between those that are displaying persistent TBI symptoms versus those that are non-TBI, and also could distinguish other elements of the experimental groups. And so what I wanted to do is take this technology and move it to a preclinical model. So in our case, it's a rat model. And the goals that I had was to minimize overt brain injury and to cause persistent symptomology. And we wanted to balance these things because you want to have the persistent symptomology, but then also to have the treatment. You wanted to maintain the substrate for repair, which would be the brain tissue. So our first model that we used was a controlled cortical impact model. This was a unilateral injury. We actually opened a window in the brain, and we actually injured the brain tissue directly. And we did this by impacting it with a pneumatic impactor. Each animal received 4 TBIs, and the parameters on the injury was a 6 millimeters per second injury, depressing the brain to the depth of 0.5 millimeter. and there was a 100 millisecond dwell time. There was 5 days between each injury, and we was 14 weeks post-injury that we did our behavioral analysis. So initially what we looked at, we noticed just in tracking the weight, as we weighed the animals on each injury day, we saw that the TBI animals were gaining less weight. And so that was a significant thing that we found during the intra-op period, or during the time when the injuries were going on. For the duration of the study, the TBI animals, the weight for them just lagged behind. It never caught up. So then when we got to 14 weeks, we started doing our behavioral testing. One test that we did was the rotarod performance test, and this is just a test that would test the balance, coordination, and physical condition, as well as motor planning, basic motor ability of the rats. It's an accelerating rod, goes from 1 revolution per minute up to 30 revolutions per minute over a 5-minute period maximum, or until the rat falls off the rod. And what we found, interestingly, is that the TBI animals actually stayed on the rotarod longer than the sham animals. And this was interesting because we would have expected that the sham would have stayed on longer. But there was an issue that we did not have a deterrent, like a shock plate at the bottom of the rotarod chamber. So it might have been that the shams did not have motivation to stay on the rod. We also looked at the elevated plus maze, which is a test of anxiety, and a fear of open spaces. And so the idea is, if the rat is suffering from anxiety, it would stay in the closed arms as opposed to the open arms. We did not find any difference in the time spent in either the open arms or closed arms for either group. We also did the open field test, which is a basic motor task, but also a level, measures the level of anxiety, whereas an animal exhibiting anxiety would spend more time up against the walls of the chamber as opposed to the center zone. And you can see here on the bottom, we also look at the distance traveled just as a measure of overall motor ability, and we didn't see any differences there. I'll refer back to that later. But in terms of the results, we did not see any significant differences in the times that the animal spent in the center zone. But we did see that the TBI animals were actually a little bit more active than the sham animals, had more active time and less inactive time. We did the force swim test, which is a basic measure of whether the animal is exhibiting depressive-like behavior, or a measure of behavioral despair. When they're placed in the water, they will exhibit escape activities, whether it be climbing or swimming, trying to get out of the water. And then when they exhibit depression, they will be immobile. And so what we found, any differences in this test either. So we did not see any terms of depression-like behavior or despair. Lastly, on this model, we looked at the Morris water maze, which is a test of spatial learning and memory. The animal is trained to find a platform. And initially, we did training trials where the platform was actually above the surface of the water, and then they were trained over 3 days to find the platform submerged underwater. And then finally, we removed the platform completely, and we looked at their search strategy to try to find the platform. We didn't find any difference in the latency of platform. So the actual maze was divided into quadrants, and We started them, and the platform was located in the northwest quadrant, and we started the rats in an equal number of times each one of the quadrants. And so, taking all those quadrants together, we did not see any difference in latency to platform. But if we started the rats in the northeast quadrant, there was the TBI animals did take longer to find the platform placed in the northwest quadrant. So, in terms of our molecular analysis, so we did RNA-seq, and this was a terminal blood draw, so right before the animals were sacrificed. We were able to find a panel of genes that could distinguish between TBI and sham, and also transcripts as well. And so, what are our conclusions from our open head model? We saw that it affected weight gain during the surgery period. The TBI animals gained weight at the same rate as controls for the duration of the study, but lagged behind. The rotarod results may be reflective of a lack of motivation for the sham rats. And we are seeing some memory deficits in our TBI group based on start position. But our model is not resulting in persistent anxiety-like or depressive-like behavior. We did see a set of genes and transcripts that can distinguish between TBI and sham. And so this RNA-seq data demonstrates that molecular disturbances are detectable even when the behavioral impact is not significant. And so, since I titled this talk "Lessons Learned," let's talk about the lessons that we did learn during this process. Lesson number one was that these rats were too big for some of our standard rat behavioral apparatuses at the chronic time point. So we were using male Sprague Dawley rats, and they do grow very large. And so it caused difficulty in interpreting the rat's observed behavior. Especially on the open field test, a rat could be near the wall but still have significant portions of its body in the center zone, or vice versa. And on the FST, it may be that these results could be invalidated because the rats had the ability to touch their tail to the bottom of the cylinder. And we tried to add more water to the cylinder, but that allowed the rat to climb out to the top of the cylinder. And so we could not do that as a corrective measure. So the solutions to these is that we increase the size of our open field chambers from 17 by 17 to 36 by 36, which allowed for a clear distinction between the center and surround zones. And the 12-inch diameter 4 swim test cylinders were increased in height from 17.5 inches to 24 inches tall, and the rats could no longer touch the bottom with their tails nor climb out of the top. Lesson 2 was that while our interest is in chronic outcomes, we may have missed some early behavioral differences. So the solution there is basically to collect behavioral data at regular intervals using tests that are not biased by repetition. Lesson learned number 3. So we were at a state here where we weren't finding significant differences, and our funding was nearing its end, and so we wanted to get an extension in our funding. So we approached our funder, and if you ask for an extension of your funding, they might ask you to change your protocol in order to give you that. So what is the solution to that? You make the change. And so the requested change was to move from the open head model or surgical model to a closed head injury model, thought to be more clinically relevant. And so how we went about doing that was, instead of having a unilateral injury, which was angled, we actually did a vertical injury midline. Obviously, we shaved the head. We did not open the skull. And we attached a silicone bumper to the end of the pneumatic piston. And then, since the lessons we learned from the surgical model, we wanted to do a pilot study so that we could kind of hone in on the optimal parameters for the injury model. So we wanted to do multiple injuries. We wanted to do 4 injuries again, but at various intervals and severities. And then we wanted to do biweekly or every 3 weeks behavioral testing. And we wanted to do this up through 14 weeks for the analysis. So the pilot study details, we used either a velocity of 4 meters per second or 6 meters per second, a displacement of either 4 millimeters or 6 millimeters, and intervals between injuries of 1 day, 3 days, and 5 days. And then we had the appropriate sham groups and a naive group. So it looks like this in terms of all the animal groups, but and basically when I go through this, if I refer to the animals, I always talk about velocity first and displacement second. So if it's 4, 6, it'll be 4 meters per second and 6 millimeters displacement, just as I go through. And so the behavioral tests that we used, we only use a test again that could be repeated without having a bias for each test. And so the tests that we used were the rotarod test, open field test, and the force swim test. And then our modified CCI device setup looked like this. We had the silicone bumper attached. We put it at a 10-degree angle so it would better fit the shape of the rat's head. They were placed on the EVA foam pad. And if you notice here, there's a nose cone, which they were kept on isoflurane until 30 seconds prior to injury. So we looked at right latency, which is supposed to be an indication of how severe the injury is. Once again, we kept them on isoflurane up until 30 seconds before injury. And just this is just the 1-day animals that I'm showing in this particular case. Initially, we were using 3% isoflurane because that's what we use to maintain our animals during surgery. And so we initially weren't seeing any difference in right latency. And we were like, well, this is unexpected. So we kind of went back to the literature and realized that maybe we should reduce it to 2% isoflurane. And once we did that, we were able to see striking differences between our most severe group, which is our 6 group, 6 millimeters per second to 6 millimeter depth, with all the other injury severities and the sham group. And so what that looks like, if we sum it all together, is that the most severe injury showed a higher right latency than all the other groups. And I guess I should go back and say that right latency is after the injury, you turn the animal on its back, and how long it takes for it to right itself or turn back over is the right latency. And so these animals, the most severe injury was different than the other groups in terms of right latency. For the 3-day animals, we saw the same thing. However, for the 5-day animals, we did not see this difference in right latency overall. So we kind of found out early on that the 1-day animals were probably going to be where we would land on in terms of moving this whole thing forward. And so when I'm showing this weight gain data, I'm just gonna, I think, just show the 1-day animals here. So after the first injury, the most severe injury gained less weight than significantly less weight than the sham, the 4-4, the 4-6 animals, but not the 6-4 animals, and that's 6 millimeters per second to 4 millimeter depth. And so, then for the other time points, there were no significant differences in weight gain, but because of the severe weight deficit and weight gain after the first injury, the overall weight gained during the injury period was less for the most severe injury, and that's shown by this graph here. So in terms of our behavioral test, we really only saw some anecdotal evidence of any differences here. So with the rotarod, this is just 2 weeks post-injury, and this is a difference between the naive group and the group that was 4 animals with a 5-day interval. They spent less time than naive on the rod. But there was a lot of variability with this, and we didn't really see any differences on rotarod. And it may not be surprising because of the location of the injury. It was a frontal injury on the brain, which may not have that much effect on the motor behavior. The open field test, now this was an interesting result because this was 3 weeks post-injury, and then the naive group, and obviously on the open field test, they were given 5 minutes to explore the open field, which is 300 seconds. The naive group spent over 150 seconds exploring, would spend time in the center zone, whereas all the other groups, including the sham groups, spent basically less than 3 seconds in the center zone out of 300 seconds. And so once we move forward in time, 5 weeks post-injury, all the animals were spending less than 3 seconds in the center zone, and we saw similar results at 7, 9, and 13 weeks post-injury. And so basically, these rats weren't spending any time in the center zone. They were basically spending all their time up against the surround of the open field. And so we weren't seeing much differences with the tests we were already using, so we wanted to extend our pilot to include a couple more behavioral tests. And so for this, we only use 6 meters per second in velocity, and we use displacement of 4 millimeters per second. Displacement of 6 millimeters and 4 millimeters, and 1 day between injuries. So that's just these groups here, and we aimed for 8 animals per group. And we introduced 2 new tests: the novel object recognition and the Barnes maze. Okay, and so we looked at weight gain again, and then our most severe injury, 6'6", gained less weight during the injury period, as expected. Novel object is you place the animal in a open field with 2 familiar objects initially and allow them to explore the objects. And then after a 1-hour delay, they are placed back into the chamber, but one of the objects is replaced with a novel object. And how much time they spend exploring the objects is recorded for a total of 5 minutes. In theory, the rats should spend more time exploring the novel object unless there's a deficit. So at 4 weeks post-injury, we didn't see any significant differences in the times that the animals spent exploring the objects. At 8 weeks post-injury, both the 6'4" and the 6'6" animals spent significantly more time exploring the novel object than the familiar object. The sham animals did not. But at 12 weeks post-injury, the 6'4" animals spent significantly more time exploring the novel object, but the most severe injury, 6'6", they seem to have reverted back to not showing any difference between their exploration of the 2 objects. So that's all the behavioral results we have thus far. We're still collecting data, and so hopefully we'll be able to share that soon. But we've also started a new collaboration where we wanted to look at visual deficits due to our model of TBI. We started this collaboration with the labs of Andrew Fiola and Rachel Allen and Amber Douglas, who was in their lab. Excuse me. So visual deficits are commonly reported by veterans who have suffered TBI, and they're usually associated with TBI caused by blast injury, which involves direct pressure on the eye. But we wanted to look at the effect of our concussive model of repetitive mild TBI on the structure and function of the eye. And so these are the results that have come out, these very preliminary, very small studies. In this case, we looked at the Sprague Dawley rats, and we did only 3 animals, sham and 3 TBI, and they were just the 6'6" animals, so the most severe injury that we've done. But we did see a difference in the B wave. In other words, the amplitude of the B wave and the electroretinogram. And excuse me, because I'm somewhat of a novice with this visual data, but in terms of the electroretinogram, we saw a reduction in the B wave amplitude, which began began showing up as early as 1 week after injury in these rats, and it continued on all the way through 9 weeks. However, with the Long Evans rats, we repeated the study, and actually we did 5 injuries on the Long Evans rats compared to the 4 injuries on the Sprague Dawley rats. And the main difference here is that Long Evans rats are pigmented. In other words, their eyes have pigment, so they see better, and so they're better for visual studies. And so in some literature, you would expect a more robust visual response. Ironically, there seemed to be a delay in how soon this difference in the B wave amplitude showed up. It did not show up until the 4th week, and it's still not yet significance because of the variability. So there's no significance here, but it starts to show a decrease in the TBI animals at 4 weeks in the Long Evans rats. So here we are. We have preliminary conclusions with the closed head model. Right latency appears to be increased for the most severe injury. Our closed head injury model affects weight gain during the injury period, except for the 5-day interval animals. We're not seeing any differences, again, for our anxiety measure or for motor function, but we might possibly see a difference in recognition memory with the novel object test. That remains to be seen. And then our injury model seems to reduce the amplitude of the B wave component of the electroretinogram, which is thought to reflect bipolar cell activity in the retina. So what's still to come? The forced swim test results from the pilot study are still being collected and analyzed. We have to look at the Barnes-Maze results from the extended pilot, which is another test of memory, spatial memory. And we have to do RNA-seq analysis on the closed head injury blood samples. And then we also want to expand our analysis of visual and retinal function. So back to the lessons. So sham animals behave more similar to TBI animals than naive on some tests. And so solutions are, if possible, we must include a naive group in our experiments just to make sure we can see what an animal that hasn't been exposed to stress might behave as opposed to the others. And so we must track the stress indicators as well to determine the impact of stress on the observed behavior of the animals. Lesson 5 is that the current behavioral tests are not revealing robust differences in this closed head injury model. And I don't have any solutions for this yet, but I have some thoughts is that, it may be possible that if we, go even more specific into some executive functioning tests, we may reveal differences between our groups. But then the question is this the preclinical model that we set out to establish? And I would say that it may be time to revisit these goals. And so our goals initially were to minimize overt brain damage and cause persistent symptomology. And so I think we've achieved goal number 1, but we obviously have not achieved goal number 2. And so do we go to a more severe injury, which then might start causing overt brain damage in order to get the persistent symptomology? And that's an open question. The issue is if our goal then is to use this model that we established for testing therapeutics, do we then remove the substrate for repair because we now have damaged tissue and the substrate was no longer there in order to execute the repair due to the therapy that we give? And so I don't have an answer, but that's where we are now. So I'll just leave it there. I want to acknowledge my lab members, Martha Graham, who basically kept our lab open throughout the the pandemic. When I was ready to shut it down, she refused and she saved our data. So I really appreciate her and all the work that she spearheaded in terms of our behavior. Patria Juzang, who's our technician, who has also done a bulk of the behavior analysis as well. The students in the lab, Dallas and Olisa, and also the Fiola and Allen labs, Andrew, Rachel, and Amber, who have helped out with this collaboration on the visual data that we are beginning, and then our funding source. So the last lesson that I want to share is that there will be times when your research is not going the way you expect or desire, and the solution here is to reach out to people you respect for their insight and suggestions. And with that, I open it up.
Thank you from the whole audience. I know that I'm the only one unmuted right now, but what I will do is, if anyone has any questions, feel free to either put it in the chat or you can unmute yourself to ask questions. While that's getting set up, I actually have a few questions, so I'm happy to start. Thanks for this great talk. Oh, actually, you know what? Michelle LaPlaca has a question. Well, I'll let her go first.
Okay.
Can you hear me?
Yes.
No, thanks, Todd, for that talk. You did a lot of different behavioral analyses, and I know that can be sometimes, very grueling to do all those. I was wondering though if there were any correlations between for particular animals in their behavioral deficits and in their vision deficits.
Right, so that's a great question. So we haven't advanced far enough in the visual analysis to compare. Actually, the animals that have received visual analysis did not receive behavior yet. That's what we've actually, as part of my grant submission, that's hopefully will be funded. And so then we'll be able to get that answer for you, but we haven't done that yet. But one thing that's important there is in our initial test in the Morris Water maze, where we saw some difference in positional starting position, it may have been an effect of the visual deficit because we had a unilateral injury, and it might have been some imbalance in the visual acuity there.
Sure, and just one more question. Did you, I know that in the closed head model, there's no overt brain damage. What about at the cellular level? Do you see any deficits in the cells and either right under the injury versus, I know you're looking at some hippocampal functions or in the hippocampus?
So that's to be determined. So we're still cutting that tissue and staining it. So, hopefully we'll have that answer soon.
Okay, thanks.
Yeah.
And anyone else who has questions, you can feel free to, again, use the chat or use the raise hand function. I'll go ahead and ask the questions that I had, which is that I was curious whether or not you think that, depending on the age when you deliver the TBI, like either in the open or the closed cell models, open or closed head models, the age at which you kind of deliver that TBI, do you think that would kind of affect, any of the results, whether it's like weight gain or the behavioral results or the RNA-seq?
Yeah, so I think it probably would. We tend to give ours in what would be considered a young adult rat, but I'm sure if we went older, it probably would have more differing effects. I can't say exactly what they would be because we haven't done it, but yeah, I would expect there would be some other difference especially if it was like an aged rat.
Okay. But, okay, I guess I can see the motivation for the young adult though, especially in terms of clinical translation for veterans.
Yeah. I mean, part of it is if we really look at from the VA perspective, most of the active duty military are young.
Yeah, exactly.
So they suffer these injuries at a young age, and then they deal with these ailments when they're older. And so that's kind of the motivation to have this chronic outcome after suffering this injury at a, say, young adult stage.
Right.
Analogous to, yeah.
And then the other question I had was, about some of these kind of counterintuitive results that you got in that open head model, like the improved rotarod performance and the heightened motor activity.
Hmm.
And I'm wondering whether or not like that there may be even some kind of compensatory response or something. I forgot exact. I know that in the closed head model you said it's a frontal injury. I forgot exactly where the injury is in the open head model.
Right. So in the open head model, it's more of a sensory motor cortex injury, and
Okay.
so yeah, so that is actually one thing that we thought in ischemic injury, there's a concept of preconditioning where
Okay.
you have a previous injury and then that causes to protection from a later injury. And so we did think that having them be 5 days apart, there might have been some element of a preconditioning kind of protection, but we haven't verified that yet. Um, so that would be something that we want to look for in the future, but we can't say definitively if that's what we're seeing.
And then the last thing I wanted to ask, this is, maybe a little bit similar to what Michelle was asking about the visual activity, but I'm curious about, you said that you have plans to do behavioral experiments on some of those animals that you started to look at the electroretinogram.
Mhm.
And I was curious if any of the behavioral assays that you have lined up are kind of visually guided behaviors that, sort of I don't know, might connect something about that visual impairment with
Mhm.
visual behavior.
Yeah, so I would think the novel object could have an element of vision. The Morris Water maze as well as the Barnes maze, which I didn't show today because we're still kind of analyzing that data, um, could have a visual element to it. In terms of the other ones, I don't know. I don't think how I think they're more of a measure of anxiety or trying to escape the water in the
Sure.
force swim test. So I don't know if vision would really come into play as much in those tasks, but in the ones where you're actually having to explore an object or find the escape, um, either platform or box, I think though the vision could be a factor because there are visual cues that are placed to guide the rat to the location of the escape.
Right.
Yeah. So,
It looks like there's a question in the chat from Johan. I don't know if I'm saying that correctly, but if you want, you can go ahead and unmute yourself and ask, or if
Sure.
you're not able to. Okay.
That's okay. Hi, it's Johan, but it's okay.
Johan. Okay.
I was gonna so I part of my PhD I did on traumatic brain injury, and I guess one thing I was wondering is we were looking at for subpopulations, and have you found any? Did you look at any of that type of analysis within the TBI group? Because we found the same thing. Like when you look at the group as a whole, like you don't see any differences. You know.
yeah, so I think, in terms of analyzing them all together as one group as opposed to breaking them down kind of pairwise or by injury day and so on and so forth. Is that what you're asking?
Um, sure. I mean, we were we were we didn't find it like that though. We didn't find any differences.
Yeah,
We
so
found it, yeah.
I'm just trying to understand exactly what you mean by
Um, because I was
subpopulations.
curious about the mRNA differences you found. If you followed,
Oh, okay. So we haven't gone that far yet with the analysis of the molecular analysis. That is something that's upcoming. Um, we have that we found those initial panel of genes, but we also have to go back and do some further analysis where we're going to be looking at novel genes and trans, and then we'll be redoing the analysis, including those, and also doing some further analysis. So I guess at that time we would probably look for, if you're talking about molecular subpopulations, I guess we'll be looking at that in that analysis, I would think.
thank you.
Yeah.
All right. Let's see. If there is there any more questions?
I can ask a quick question. Hey, Todd,
Great.
thanks for joining us. It's nice to see. We like to use the seminar series. I used to run the seminar series before Ming and Toby took over, and it's really good to see all the young neuroscience-related faculty in Atlanta getting together. So thanks for joining us.
Absolutely.
And I'm sort of totally in a different area of neuroscience, and I sometimes like to get a sense of like what with these translational approaches. You made a really interesting statement to me that your program officer had encouraged you to switch to a different model system for TBI, still in the rat, but with a different a closed head, I think you said. I was
Yeah.
just curious what sort of in a general sense, for the uninformed of us like me, makes a good traumatic brain injury model. I'm a little unclear about like at the high level, what are the issues that are going into. I'm sure there's no like perfect answer to that, but like what are the issues that you're considering when you're making a pick between these different models? I think that might be sort of fun.
I wish I knew the answer to that because that's part of where we are. Specifically me, but in a larger scale, the field is what are the proper parameters for a translational preclinical model. I guess it depends. A lot of depends on what you're looking at, and what your goals are. I mean, I shared my goals, and I'm having trouble achieving those at the moment. But other people are looking for different things, and so they might have a more. It might suit their purposes to have a more severe injury, right? And so they don't mind having cavitation of the brain and those type of things. I was trying to avoid that. And so there's multiple models. I thought our surgical model, I still think has great validity to it, but you know, the lesson I learned there was that, in order to maintain my funding, I had to switch. And so there's the theory though that the closed head injury, which happens more often, um, clinically is more relevant, and so it's good to look at this closed head model, but it's just we have to figure out how to get to the place we want to be with it in terms of the parameters. So I don't know if that answers your question.
That was helpful, yes. And Michelle in the chat just said that she was telling me that modeling TBI is sometimes called the Wild West. It looks like that's true experimentally too. So,
It sure is.
Hi, Todd. I guess I had a sort of similar question to Simon, but maybe just slightly different. So, are there other models from other animal species, like maybe larger animals,
Yes.
TBI that are more successful, more accepted?
Um, More successful. That remains to be seen. Um, there are larger animal models, and the brains of those animal models might be more similar in terms of their structure, to a human brain. Um, but we're all kind of working towards trying to find things that work, that are translatable. And so, um, I think, I think, for what I'm trying to achieve, it might be fruitful to look for one of those other models because you might be able to get the unique thing about a human brain is that you can have an injury that doesn't show up so blatantly on like a CT scan. It'll show up maybe on an MRI or a specialized MRI, but you'll have these severe deficits. In the rat, it just does not appear that that's what we're going to get. And so I don't know what they're seeing in, say, like the pig model or whether they're seeing, um, this where you can have very mild structural disturbances and still have severe or persistent symptomology. So I look into that, and then we'll see if I can get funded to look at that. I'll definitely do it.
I guess one thing that I'm just trying to think about. So, there's many types of damage to the brain, right? There's obviously just gray matter versus white matter damage, and I don't know, different layers of cortex or different types of subpopulations or something. Uh,
Hmm.
is it possible that what you're doing to the rats is just damage, but of a different kind than what humans are suffering. For example, like maybe humans get white matter damage and you mostly do gray matter. I mean, just shooting in the dark.
Yeah, I mean there are different injuries. I mean, if you look at um, diffuse injuries, which is a closed-head injury, is considered to be a diffuse injury, and so there are different. You don't have the focal injury. Like when we were doing our surgical model, we had a very focal injury, right? It's localized to that one spot. We can compare the ipsilateral side of the brain to the contralateral side of the brain. But where we're hitting it midline and frontal, but that force is transmitted throughout the whole brain. And so it becomes a diffuse injury. And so you'll get both, a shearing force, which can cause damage to the white matter tract as well as gray matter. And so but we're still in the process of doing our histology, so hopefully we'll get an answer as to what our particular model is doing to the brain. But yeah, and what parts of the brain are most affected are going to affect what behaviors are appropriate for studying that particular model. And so I think once we get our histology, that may help us kind of tailor what behavioral tests we use. But then I also go back to what I said in the talk is that I set out with a goal of really looking for a preclinical model that was having, particular, I wanted to see anxiety-like behavior and depressive-like behavior persistent to a chronic time point. And so by just finding another behavioral test may not address that particular goal, which was to have that particular preclinical model. And so, there's two things warring, right? But my father-in-law has this saying is like when you reach a fork in the road, take it. And so, can we go in both directions at the same time? Then that's possible, and then we can maybe get more bang for our buck that way.
Cool. Yeah. Really important work and really, really interesting.
Yeah, are there any other questions that people have? They can feel free to unmute themselves. Okay, well, this was actually a great discussion. This is like exactly what we're looking for to have, a wonderful talk like this, but to generate a lot of discussion from, people who are really immersed in the field versus, kind of people outside of it. So I'd like to thank Todd again for coming to give this great talk,
Thank you.
and thank everyone for joining us.
I appreciate it.